The two different colored chromosomes represent maternal and paternal chromosomes. The purple is paternal and the green is maternal.
In metaphase of mitosis, all of the chromosomes are aligned at the equator of the cell called the metaphase plate. The microtubules that allow the attachment of the chromosomes to the mitotic spindle are what pull the chromosomes to the center of the cell. This ensures that each cell will contain one sister chromatid from each of the duplicated chromosomes.
Anaphase begins with the degradation of proteins that hold sister chromatins together, freeing the individual chromosomes. The freed chromosomes are then pulled by their centromeres to opposite poles of the cell. Other spindle fibers lengthen to push the poles farther apart to elongate the dividing cell. By separating the replicated chromosomes, this ensures that the two new daughter cells will have an identical set of DNA material.
In metaphase 1 of meiosis, the paired homologs align down the center of the cell. The purple chromosome from one parent is lined up with its green homolog from the other parent. The alignment of chromosomes during this phase is a random process, meaning that there may be a paternal chromosome on the left and a maternal chromosome on the right. For the next homologous pair, the positions could be reversed. In the case of n equals 2, there will be 2 to the second or 4 unique chromosome configurations. This is known as independent assortment.
In anaphase 1, the homologous chromosomes separate the opposite poles of the cell. However, the sister chromatids remain stuck together. The homologous pairs are not necessarily identical, so that when the homologous pairs are separated, it encourages genetic variability.
In metaphase 2 of meiosis, the chromosomes align along the equator of each of the two cells created in meiosis 1. The mitotic spindle pulls the chromosomes to the center of the cell. Their alignment at the center will ensure that when the chromosomes split, each cell will produce 2 haploid cells for a total of 4 haploid cells.
In anaphase 2, in two separate cells, the centromeres split as the sister chromatids separate to opposite poles of the cell. The chromosomes are split to ensure that the haploid cells each have half the amount of DNA.
The purpose of mitosis is to yield 2 clones or 2 identical copies of the parent cell. This process is used for growth, development, and repair of cells in an organism. It is much faster and uses less energy. 100% of the genes are passed on from the parent cell to the daughter cells. There is no variation in mitosis. The only variation comes from mutations, which are not always good.
The purpose of meiosis is to create genetically different sex cells that will eventually fertilize another sex cell to create a new organism. Meiosis halves the chromosome number of gametes so that the number of chromosomes doesn't double with every generation. I am now going to talk about two different types of variation. One is crossing over, which is during prophase 1 when the homologous pairs come together. The pairs sometimes cross over and switch tips, which causes genetic variation. The other one is an independent assortment, which is another type of variation which occurs during metaphase 1. It is determined by the way in which the homologous pairs line up. In meiosis, homologous pairs align, which does not occur in mitosis, which allows for crossing over, which also does not occur in mitosis. Also, in meiosis, one cell creates 4 daughter cells, which is different than in mitosis, where one cell makes 2 identical daughter cells. In meiosis, there is a lot more variation that the daughter cells could have, which in mitosis, the only way to have different DNA is through mutations during the S phase of interphase. Also, another difference is that in mitosis, it only occurs in somatic cells throughout the body, while in meiosis, it only occurs in germ cells. Also, in cytokinesis, it only occurs once in mitosis, while it occurs twice in meiosis, once in meiosis 1 and a second time in meiosis 2. In mitosis, the cells that are produced are identical, while in meiosis, the cells produced are non-identical haploid cells.